Hello, everybody. I think we wait a little. We wait enough for everybody, for all the attendance. So we are going to start with the presentation. First of all, I want to thank you for attending to this webinar. And I am going to introduce myself. I am Jose Miguel from Condalow. I am the product manager of bioprocesses. And today we are going to talk about the Condalow peptones, their effect in the biopharmaceutical industry, and the main cell lines used for the manufacture of biological drugs. Without more delay, we are going to start with the presentation. Okay, I think now you can see my screen. So first, here we have the index, so we can organize our ideas. First, we're going to talk about the bioprocesses in the pharmaceutical industry. We're going to give a little of explanations. Then we're going to talk about how to produce these biopharmaceutical, these biological drugs with the cell lines, the culture media, and finally with the supplements, in this case, the Condalow peptones from Condalow. And finally, we will get to the conclusion of this PowerPoint, of this presentation. Well, most medication we find are made of simple molecules. These molecules have different therapeutic effects, and they are capable of deceive the body's receptors to generate a response, a therapeutic response. These elements are obtained mainly through chemical synthesis, obtaining different compounds to stimulate certain reactions and obtain these active molecules. It has been the main path of the pharmaceutical industry until the second half of the 20th century, where the advances of the biology, the genetics, and the biotechnology made it possible to identify the effect of the main regulatory proteins and which genes encodes them. With this information, we achieved to produce the first recombinant protein, the recombinant insulin, in 1982. This drug was before obtained through the purification of the porcine pancreas, but when the insulin recombinant insulin arrived to the market, it generated a great difference because it was obtained through the inserting of the human insulin gene into a plasmid and then introduce it into the strain of E. coli. This gave a product much easier to produce and without a risk of immunogenicity. Since then, the industry has begun to synthesize these biological drugs, medication with the same structure as the molecules of our body. These molecules are produced through microorganisms or living cells. The industrial procedure to obtain these products using the cell cultures is called bioprocesses. According to the WHO, the World Health Organization, biopharmaceuticals are medicines made from materials from biological origin, such as microorganisms, organ tissues from plants and animal origin, cells and fluids from humans or animals, and biotechnological cell design. This definition includes the vaccines, the enzymes, plasma and blood derivatives, allergens, antigens, immunoglobulins, hormones, cytokines, and diagnostic reactives, and some diagnostic reactives. But the pharmaceutical companies focuses especially in the drugs that they can produce in large scale with the bioprocesses. These will be the recombinant proteins. Some examples of the most famous recombinant proteins are the monoclonal antibodies, hormones, or coagulation factors. It is estimated that biopharmaceutical market generates over $355 billion globally in 2021. And it is hoped that in just 9 years, in 2030, it generates over 7,000 billion of dollars. This is an increment of 144%. But why are the pharmaceutical companies investing so much in this kind of products, in the biological drugs? There are two main reasons. The first one is that it is very difficult to find new active molecules. It is needed to realize massive screening just to find new candidates, and the options are more and more limited each year. On the other hand, the biological synthesis is booming. It started in the decade of the 80s, and now we are just starting to discover this new field. At this moment, there's no sight of limit to the option of these kind of drugs. Another argument is the effectiveness. The biological drugs are molecules with the same structure as our own proteins. It means that their effectiveness is much higher with a greater therapeutic capacity and more specificity. We can say that these drugs are using the same language of our own body. But in order to synthesize these drugs through bioprocesses, we need the specific elements. That will be the expression system or the cell lines, the culture media, and supplements to enhance the effect of the media. The expression system or the living cells using in the bioprocess are a key point to synthesize these biological products correctly. There are many kinds of expression systems. For example, microorganisms like bacteria or yeast, or eukaryotic cells like insect, plant, and mammal cells. Usually, the industry use mammal cells. Also, I want to specify that the cell line term refers to the manipulation of any kind of cells. However, it is commonly used just for the application of mammal cells. But why does the pharmaceutical industry use the mammal cells? Well, these kind of drugs began to be synthesized in microorganisms, such as the aforementioned example of the recombinant insulin. Also, the mammalian cells present some challenge respect microorganisms, like they are pluricellular beings. Their cells are not independent and they form a part of whole. This generates an important problem. It's difficult to grow them in vitro. Despite this main problem, the mammalian cells possess a key advantage. They possess post-translational modification. Proteins synthesized in mammalian cells, such as human cells, undergo modifications during their biogenesis. These changes can be the one we can find in the screen: the phosphorylation, methylation, glycosylation, ubiquitination, and acetylation. The microorganisms lack the ability to produce these modifications, so the recombinant protein will be inactive in most cases. If we want to produce drugs, biological drugs, functional to the human beings, it is required to use expression system similar as possible to our own cells. So we already decide that we have to use mammalian cells, but which one? Also, we don't use any cell that we catch from a patient. We are using what is called cell lines. These are populations that are culture of animal cells with the ability to prevent the senescence, giving them the ability to duplicate repeatedly. They do not lose, they don't lose their genetic code doesn't suffer damage between replications. The industry uses many kinds of cell lines with different qualities that favor the synthesis of recombinant protein or other products. The most popular are the following ones: are the CHO cells from Chinese hamster. They are specifically used for recombinant proteins. And S0, they come from a myeloma, and they are used for the for synthesis of monoclonal antibodies. BHK21, they are used for when you are working with virus. It could be vaccines or viral vectors. Hybridome, specifically for monoclonal antibodies. These kind of cells are the fusion of a myeloma and a B cell. SP2 cells also comes from lymphoma, like NS0. And finally, HEC, this kind of cells comes from human kidney, and they can be used for the synthesis of recombinant proteins, vaccines, or even viral vectors. Here in this graphic, we can see the different application, the different use of these kind of cells, of this type of cells in the industry. As we can see, CHO cells is the most popular cell line. Some companies also use NS0 and SP2 because the main kind of product are monoclonal antibodies. But without any doubt, CHO is the king of the cell lines. But why are CHO cells so popular? Well, these kind of cells were obtained from Chinese hamster ovary in 1969, and since then they have been widely used in the research industry, in the laboratory research, and then rapidly became the favorite cell line in the synthesis of biological drugs. Its popularity is based on these following qualities. The first one, they present a greater resistance to change in the environmental, in the culture media, such as alterations in the pH, in the temperature too. Also, they can grow in suspension culture, favoring the use in the bioreactors and the scale up of the bioreactors. They are one of the cells with the highest rate of recombinant proteins production. Also, they are not human cells, they are hamster cells, but their post-traductional modifications has a resemble to the humans. So the product produced in these cell lines has a low immunogenicity. And finally, finally no. There's also a wide range of chemically defined media for this kind of products. There is a lot of alternatives. In the next step, in the next page, we will talk about the different kind of culture media. And finally, they have a low mutation rate. They have a high genetic stability. So after multiple divisions, replications, they maintain their genetic code with little variation, with little modification. Now we are going to talk about the culture media. Excuse me. The cell lines are a very sensitive type of culture, so they need a specific cultures media that works as a substrate where they can grow. They also give the protection from the environment. They create an environment that take care of these cells. They are a constant supply of nutrients and also eliminate the waste of their metabolism. All of these requirements are given by the culture media, this liquid solution with all the factor and nutrients to stimulate the growth and the synthesis of products of biological drugs. The main elements of this culture media are the one we can see here. Firstly, the buffer, for example, the sodium bicarbonate, which regulate the pH of the solution. Inorganic salts like sodium, calcium, or even potassium. These ions are essential for the membrane electric potential. Then we have the amino acids. They are the bricks of the proteins, and they are needed from the growth and specifically for the synthesis of products. I want to highlight the L-glutamine because this amino acid has a low chemical stability, so it decomposes rapidly in the media, and usually the dust add this amino acid just before the application. Then we have carbohydrates. The most famous is the sugar, and is the main source of carbon and energy. Then we have lipids, such as cholesterol, required for the cell and other molecules structure. We have the trace elements, metals used as cofactors in enzymes, in essential enzymes for the metabolism. Finally, vitamins. These organic and inorganic compounds that the cells can produce by themselves. There are many kinds of culture media, and they can be used one or other depending of the cell type, and also depending of the metabolism, the product of interest you want to produce. There is a great difference according to its composition, but the most determining and defining values tend to be the variability and the presence of animal origin compounds. Depending on the composition, it can be classified in basal media with serum, serum-free media, and chemically defined media. Basal media is the best known and the first to be developed and used. There are many kinds in this category, but all of them have something in common, and it's the dependence of the fetal bovine serum. This supplement is obtained from fetal bovine, and it was first used in 1958. And since then, it was the main additive, the main supplement in culture media. It is due to it can be used for any kind of culture media or cell line, and has a high performance in the density of cells and the yield of product obtained. But this supplement has serious disadvantages. For example, there's a high variability between batches. There is a high risk of contamination, which include bacteria, virus, or even prions. And finally, is the ethical problems to obtain them. Following the basal media, we have the serum-free. This kind of media do not require the FBS, the fetal bovine serum. They are usually made of extract or hydrolysate from different sources. It can be animal or plant source, and can be classified depending on the presence or absence of whole proteins, and if there are fragments of them, the maximum size of these fragments. The main advantage is that they don't use fetal bovine serum and has a lower price, but a serious disadvantage is that they don't usually have the same performance as the other culture media. Finally, we have the chemically defined media. Nowadays, this become a very famous kind of culture media because the composition is completely known. There is no source of variability in its production. Additionally, these media usually don't contain animal origin components. The main advantages of this product is that they don't possess variability, and it's easier to purify the biological drug, the biologic product. But the main problem with this kind of product, of this kind of media, is that it has a high specificity for the cell line and for the product you want to produce. You can't use a chemically defined media for different processes or different cells, and it usually has a higher price. Also, the chemically defined media usually need more supplements than the serum-free media. As I said before, media are usually supplemented with different compounds. These additives, called supplements, could be for example, the fetal bovine serum, or many others with different effects. These supplements gives more flexibility to the culture media. The main kind of supplements will be the fetal bovine serum, the antifoam. This in bioreactors, the high revolutions can generate a lot of foam, and this can inhibit the exchange of gases. Also, there can be used as pH indicator. For example, the red phenol, which is very used in the culture media, giving this famous red color. Then there is media regulators, other kind of supplements that helps with the accessibility of the nutrients. And then there are so many kind of supplements that can be used as nutritional support. to enhance the growth and the synthesis of products. One of the biggest problems with the culture media and with the supplements that the pharmaceutical industry have to face are the endotoxins. The endotoxins are lipopolysaccharides from the gram-negative bacteria membrane, the bacteria wall, and it has a high pyrogenic capacity and suppose a great danger in the synthesis of biopharmaceuticals. This is due to their ability to inhibit the cell growth, reducing the yield obtained in the bioprocesses. Also, and the most important, they contaminate the final product, which most biopharmaceuticals are administered parenterally with an injection. So if a patient receives a high concentration of endotoxins, they may suffer a strong immune response. Even they can suffer a septic shock, followed by a vascular collapse, a cardiovascular collapse. Eliminating this kind of product, eliminating this endotoxin, is a serious problem because they are not living beings. They are fragments from bacteria. So the most popular sterility methods, and for example, the autoclave is not working. So usually what companies use to eliminate the endotoxin from the material, from flask or other material of the lab, is the spirogenation process, which is more than 1 hour at 200 Celsius degree. In Fahrenheit, it is 200 Celsius degree and 400 Fahrenheit degree. Trying to eliminate these endotoxins after the bioprocess, after the purification, means the destruction of the product. So the best way to avoid the introduction of this endotoxin in the bioprocess is to use media and supplements with the lowest possible concentration guaranteed. That's where we began to talk about the Condalow peptones. Peptones are the result of the hydrolysis of proteins after an enzymatic or a chemical process. With this process, we obtain the amino acids on smaller peptides. Peptones work as source of nutrients. We talk about the different kind of supplements with, in that case, this kind of supplement work as a nutrient supplier. Also, it generates a protect effect over the cells because it helps to regulate the pH and also protects the biological drugs because they create an environment that maintains their native conformation. Also, the peptones are one of the cheapest supplements with this kind of effect. Peptones usually have a high level of endotoxins, but as we said before, they have to be eliminated for the safety of the cells and mainly for the safety of our patients. So the main method to eliminate the endotoxins that other companies use is the ultrafiltration. With this method, they ensure a concentration above over 1,000 or 500 units of endotoxins per gram. But also, eliminates the larger peptide fragments, resulting in a loss of the nutritional value. In Conda, we want to avoid this problem, so we start an intense investigation to obtain a peptone with a low level of endotoxin, but maintaining all the peptide profile. And at the end, we produce a Condalow, this kind of peptone, without using ultrafiltration. This product was specifically designed as a supplement for the manufacture of biological drugs for the pharmaceutical industry. It demonstrates a good effect in CHO and HEC cells, and its main properties are a low level of endotoxin, above 200 units per gram. This is below most of the other ultrafiltrated peptones. It also maintains all the growth factors due to avoiding the ultrafiltration. We maintain the larger peptides. These more nutrients, these with higher capacity, higher nutritional apport, enhance the growth and the growth of the cells and the synthesis of proteins. And also it shows low variability between batches. Here I want to show you about some experiment we realized in our lab. These graphics can be downloaded in our page. If you follow the AP notes, and here we can see the production of immunoglobulins in CHO cells. We used a 30 milliliter flask with a chemically defined media. This kind of peptones, the Condalow, are specifically good. They are very good in chemically defined media. And we saw that we produce even 30% more of immunoglobulin. Then we ran another experiment with HEC cells to see the density of the cells due to HEC cell line are used. They are more used for viral vectors and virus process. In this industry, the density of cells is more important, and we saw that supplementing the media with the Condalow peptones produce almost twice of the cell density. Regarding the last webinar, here we can see the different source of material for Condalow peptones. It can be from soy, from casein, from meat. Regarding our last webinar of this line, we have developed a new kind of Condalow peptone that will be Condalow wheat. This product, this peptone, was also tested in CHO and HEC cell lines, and we saw that it stimulates over 70% more of density in CHO cell, but in HEC it has an special good effect. We can highlight that this new wheat peptone is specifically good for HEC line. As we see, it produces over 150% more of cell density. Finally, we only have to talk about the conclusions or a little summarize of this presentation. The first will be the growth of the biological market how the pharma companies are focusing more and more in this kind of product each year. And as I said, it started in the 80s decade, and the patents of this product, of the first products, are starting to run out of time. So there are starting to appear what is called the biosimilar. It will be the generics of the biological drugs. Then other topic we saw was the predominance of the cell line, use over the 70% of the bioprocesses in this industry. Then we also have the relevance of the culture media and the supplements. Depending on the media and the supplements, usually depend on the cell line and the product we desire. And finally, the effect of Condalow peptones over the synthesis of these drugs and how we can obtain a higher growth and a higher yield of product using this kind of supplement. And it will be all of this. So I only can say thank you for your attention. And if you have any doubt, this is the moment to do it. We are running out of time, so I'm gonna answer just a couple of them. For example, we have, what is the anti-foam agent supply? The anti-foam has the effect to eliminate the foam of the culture media. What's the anti-foam? I don't exactly what you want to mean with the. I don't know if I think I answered your question with the use. No. Well, sorry. I will send you all the answers to the question, so no one will leave without doubts. Also, we have other question. What type of culture media Condalow provide for cell lines? At this moment, we are developing a culture media, but we don't have a specific one. We only have these supplements, but we are working for new types of culture media. And other question here we have, which is the optimal concentration for the Condalow. We usually work between 1 or 5 grams per liter. And if you use a higher densities of this product, usually has negative effects. So the best concentrations are between 1 and 5 grams. And we are running out of time, so the next question will be sent to you via email with the record of this meeting. So I hope you enjoy this presentation and it helped you. Have a nice day.